right, ladies and gentlemen, let's go ahead and get started. You should have already listened to the section that was on prokaryotes. So the gene regulation in prokaryotes, where we talked about the lac operon and the trip operon and the cap, and talking about whether those were negative or positive control. Now we're going to talk about eukaryotes. Eukaryotes are way more complicated. We're going to take a look at 5 different levels of control and kind of be introduced to them, but not be able to really fully understand them. Just getting introduced to the concepts of how eukaryotic cells and their DNA are controlled. Okay, so eukaryotes regulate gene expression and therefore their metabolism at every step of protein synthesis, from pre-transcription to post-translation. So there's different ways of controlling the genes the entire time. Now, it's important that transcription and translation in a eukaryote have been decoupled. Remember, in a prokaryote, they can all happen sort of simultaneously because there's not a nucleus and everything's small and concise. Here, they've been separated, and so there's a lot more options for controlling when the genes get turned on and when the genes get turned off. So let's start. So the first one we want to talk about is DNA access. Only keep the necessary genes accessible. So you already learned about the shape of a chromosome and how it's folded up. You learned that, I don't remember, chapter 14, I think. So it says here, in eukaryotes, DNA is wound around histone proteins. The addition of acetyl groups to histones causes them to become less tightly packed. So as they unwind, they are more available for transcription. So RNA polymerase can squeeze in and make some mRNA. I will mention that they only say acetyl groups here. So acetyl groups unwind, methyl groups rewind. So methyl groups cause them to wind up and become inaccessible. So there's 2 types of chromatin. Heterochromatin is more tightly packed, so that has more methyl groups, and it's inaccessible to RNA polymerase. It can't get in there because it's so tightly wound up. Whereas euchromatin has been unwound primarily because it has a lot of acetyl groups, and that does allow RNA polymerase to come in and to read the gene and to make the mRNA. So, and then interestingly enough, histone acetylation and methylation appear to be heritable. So we know we can inherit nuclear DNA, we know we can inherit mitochondrial DNA, but it turns out we can also inherit the acetyl and methyl groups that are unwinding and rewinding our DNA, and that becomes important in all kinds of crazy circumstances. Things that aren't totally controlled by genetics. For example, schizophrenia. The acetylation or methylation of genes that lead to schizophrenia can determine whether somebody develops schizophrenia. So it is possible for identical twins, one to have schizophrenia and one to not, and they think it has to do with whether the genes have been coded with acetyl groups or methyl groups, whether they're open or whether they're closed up. All right. And I think that's all the information that we need to know from this section. I'm going to scooch ahead. So section number 2, this is probably the most complex of the 5 ways of controlling genes in eukaryotes that we're going to come across. So this is pre-transcription. So this is control that happens before the mRNA actually gets made. So it says all the actors need to be present for the play to begin. So we have talked about some things, and I'm going to kind of scooch in. I want you to study these diagrams. Down below, it says eukaryotic genes interact with many upstream regulatory elements. So they're highlighted here: proximal control elements and distal control elements. The word proximal means close, and the word distal means far away. So some of these distal control elements, we're going to talk about them at length, called enhancers, might be thousands of nucleotides away. And so they're wicked hard for researchers to study because they affect things that are thousands of nucleotides away. And how do you know which ones affect which? And we're going to look at a little experiment to see how they've studied some of this. So it says these are DNA sequences that precede a transcription unit that need to have specific proteins present for RNA polymerase to begin transcription. So just taking a look at this is the DNA here. Here's the promoter, which tells the RNA polymerase this is where you begin. And the beginning of transcription starts here. Remember, there's exons and introns that need to be spliced out, which we'll look at a little bit later. And don't forget the addition of the methyl cap, the 5' cap, and the poly A tail, which is leading to our mature mRNA. But we're more concerned right now with these proximal and distal control elements. That's what we're going to be looking at right now. So the proteins that mediate or control RNA polymerase are known as transcription factors. So we already saw this, and I don't think I have access to it right now, but you may or may not remember when we first learned about transcription in the Prezi that was about called the central dogma. When we first learned about how transcription is initiated, we learned that here's our guitar pick RNA polymerase. It's not going to really attach until these general transcription factors, which are little proteins, they look like grapes or something in this diagram, they attach on. So here are the little ones that we've already learned about, these light purple ones, and they attach onto the promoter and then allow, and also the Tata box, don't forget about the Tata box, I love saying that, which is a repeating section of TATA, that's part of the promoter. Anyway, these general transcription factors, they have to be present in order for the RNA polymerase to latch on. We already learned about them. Now we're going to add what are called mediator proteins, and we're going to make this a little bit more complex. So possibly super far away, thousands of base pairs away, are genes that are called enhancers. And these are found in the DNA. And the enhancers can actually attach to molecules that could either be activators or they could be repressors. So in this diagram, the molecules that are present are activators. And as you can imagine, an activator is something that activates the mRNA or the RNA polymerase and activates transcription. We could have repressors in this diagram, in which case repressors would stop the transcription from happening. But we're going to look at the activators. So if activators are present, they click onto the enhancers that could be a long ways away. And now there's going to be this DNA bending protein. You don't need to know the name of it. Just be aware that the DNA can bend over. And even though this enhancer is thousands of letters away, we can bring it really close to the promoter. Look at the similarities in the diagrams. So here's your enhancers. They're yellow. There they are right there in this diagram. Here's the promoter with the Tata box. It's green. Here's the promoter with the Tata box. It's green. And then the gene that needs to be transcribed is in dark blue. So notice that the enhancers can be brought really close to the promoter once the DNA is folded over. So now if these proteins are present and the activators are present, the activators actually bond with the proteins and they form this complex that increases the amount of transcription that's going to happen. So they activate or increase the amount of transcription. So that's how they control. Remember, controlling gene expression is about either turning it on or turning it off or even super speeding it. And so these activators are making sure that it's on and moving very efficiently. All right, so here's an example. That was sort of general. This is going to be a really specific example of activators that are working. You have to remember the big picture. You have 300 trillion cells, and in every single one of your 300 trillion cells, you have all the DNA in one cell. You have all the DNA for every cell type in your body. So my cheek cell, I keep saying this, my cheek cell has the DNA for building a liver. It has the DNA for building a kneecap. It has the DNA for building a brain. It has the blah, blah, blah. So how is it that my cheek cell is differentiated into a cheek cell and not a liver or a brain or a kneecap? Why is it a cheek cell? And that's what we're talking about right here. And I'll read this to you. Control of transcription factor availability is one of the major ways that the cells of a multicellular organism accomplish differential gene expression, which in turn allows cells to differentiate, which just means that you can have from one package of DNA, you can form skin or liver or any other cell in the body. So then they can serve different functions in the organism. Okay, so let's give you an example of how these enhancer molecules that are, or enhancer sections on the DNA that are super far away, how they can control transcription of just special genes. So take a look at this one here. This says it's in the liver, it's a liver cell nucleus. Notice the activators that are present. The colors matter. So we've got a yellow one, we've got a couple yellow ones, a couple gray ones, and a couple red ones. Those are present in this particular cell. Well, it turns out that they are attracted, they clamp on to the enhancers that are a long ways away from the albumin gene. And the albumin gene is a supercritical gene in liver cells, that is. So because we have these activators present, they snap onto the enhancer before the albumin gene, the DNA folds over, and we start transcribing the albumin gene. Notice, however, the next gene, which is also present in the same cell. Notice the crystalline gene, the activators, which are in this case orange, gray, and pink, they're not present in this cell. So the protein crystalline, which is key to forming the lens of your eyes, don't you love the name crystalline? I think that's so cool. That doesn't get formed. So you're not going to get lenses in your liver cell because the activators, the molecules that trigger it, aren't present in that environment. Let's flip cells. So this time we have a cell of your eyeball, the lens cell. This time the red and the yellow activators, they're not present. So they won't trigger transcription of the albumin gene because that belongs in liver and not in your eyeballs. However, the pink, the orange, and the gray are present in the eyeball, and that causes the cell to the DNA to fold over and allows this super great transcription complex, transcription initiation complex, and allows the crystalline gene to form. All right, I wanted to show you one other thing. We've been talking about different experiments. I have to get out of this and see if I can find what I need here. We've been talking about different experiments and how important it is to the College Board that you be able to interpret the meaning of different experiments. This particular experiment is found on page, I think it's 303 in your textbook. And I just thought it was similar in many ways to the Beetle and Tatum experiment that we talked about earlier, about the one gene, one enzyme experiment that they did with mold. So it made me think, I wonder if you guys could figure out this experiment. So let's take a look. In this experiment, scientists are trying to figure out which are the control elements that control this particular gene that we're going to call the reporter gene. Don't ask, it's not important why it's called. I'll tell you just really quickly. It's called the reporter gene because it's easy to measure how much mRNA this particular gene produces. So it's easy to measure the amount of mRNA. So it's called a reporter. It's something the scientists can use to report their statistics. Okay, so they have some hypotheses that 1, 2, or 3 are controlling this reporter gene. And so we start out without changing anything, and we look at how much mRNA is produced, and we say that's 100%. When nothing is being deleted, we say that's 100% of the mRNA. That's its normal level of mRNA. So then the scientists tried deleting one of the tried damaging, just like Beetle and Tatum did with X-rays. They tried damaging one of the enhancer molecules at a time, or that's a funny way of saying it, enhancer. I don't think they call it molecules, enhancer genes at a time. So they're getting rid of these sections of DNA one at a time to see the impact. Now, if you notice, when they get rid of number one, the amount of mRNA suddenly skyrockets to 175% of normal. So it means more mRNA got produced when they got rid of that particular enhancer. The second one, we still see an increase in the mRNA, not quite as much, but it's being produced more when that gene goes away. And then with the orange, number 3, we see that the amount of mRNA decreased. So the first question that the textbook asks you is whether there is any evidence that, in fact, the scientists were right. Let me have a quick side note. Remember that these control elements are so far away, thousands of letters away. It's really hard for scientists to know. So this is the only way they can figure it out. It's process of elimination. Delete this part of the DNA and see what impact it has. Now delete this part of the DNA, see what impact it has. No impact at all? Well, we were wrong. If it has an impact, maybe we were right. And that's my hint to you. If there's no impact at all, they were wrong. If there is an impact, maybe they were right. So they've got 3 parts of 3 enhancers that they are suspicious are controlling this gene. So were they right? Is there evidence that these control elements were in fact controlling this particular gene? I'm going to give you a moment to think about it. And the answer is yes, for all 3 of them. So the first thing we want to ask is, do any of them, this is what the textbook asks you, do any of them seem to increase? Does the deletion of one of them seem to increase the amount of mRNA that's being produced? And the answer is for sure. When we delete 1 and when we delete 2, we see an increase in the amount of mRNA. So what does that mean 1 and 2 were actually doing when they were functioning if their deletion increases the amount of mRNA? Hopefully you have it in your head. It means that both of them weren't activators, they were repressors. So repressors attached onto them, and it slowed down the amount of transcription that was happening. However, if you look at element number 3, enhancer number 3, we see that when we delete that, the amount of mRNA actually does go down. So when it's deleted, it decreased. So what is element number 3 really doing? That control element is an activator. That one normally increases the amount of transcription that's happening, but when you delete it, the amount of transcription reduces. Okay, so just an example of an experiment that you ought to be able to interpret. Let me go back to Firefox, and hopefully, sometimes my video freezes. Hopefully it won't freeze this time. It did not freeze. Good. All right, we are ready to move on. The next sections I think are a little bit easier. We have totally already talked about post-transcription. So once the mRNA has been made, now we can control about how much DNA is, or how much mRNA is going to be made by changing the transcription. That's not a funny way of saying it. By alternatively splicing it. Oh my gosh, awkward words here, sorry. So remember that one gene, when we make our pre-mRNA, the introns get cut out. I'm going to show that over here. So the introns get removed and the exons get put together. But remember that the exons can be put together in different orders or in different manners. And so that actually leads to different proteins. So we can control how many proteins or how much a gene is being transcribed by controlling how the exons are put together. So that's called post-transcriptional. See if I can get it back here. Post-transcription control. There we go. Post-transcription control. All right, I'm going to skip down to number 4. This never works when I skip. It always jumps around. Sorry, it's going to be bad. Hold on. Wait till I get to the replay. Sorry. Okay, pre-translation. So this is what could happen before a protein gets made. So pre-translation, we're trying to control how much the protein products are being made. Remember, that's the whole idea. We're controlling gene expression. So we've done all kinds of things before the gene has been translated, and now we're doing it right before we make the protein. So this is an area that we know very little about, including me personally. I know very little about. These are all the different types of non-coding RNA molecules. We are just discovering these, and Wikipedia lists a bunch of them, and that was from 2011. There's way more now. So we're just going to focus on one example just to be aware of it. Remember that we have said that your DNA only 1.5% codes for proteins. 75%, and that's our latest information, codes for RNA. And a lot of these RNAs are in charge of controlling the genes, controlling when the genes get activated and when they don't. So this is one example. This one's called RNA interference, and the example that we're going to go with more specifically is microRNA. So this is what the primary microRNA transcript looks like. The mRNA, I'm sorry, it's not mRNA, it's miRNA, folds over on itself and it forms little hydrogen bonds. So usually this is single-stranded. mRNAs are single-stranded, and that's what you see here in the diagram in red. Those are single-stranded. But the microRNAs actually fold over on themselves, and they form these little hairpins. A dicer molecule comes through and cuts them off from the others. And then this is where it gets a little bit complicated. So it says the miRNAs are produced after the transcript for them is cleaved into fragments by a dicer protein. So this dicer protein made these little sections of microRNAs. They then, who knew this was a verb? They complex with proteins. So the microRNA then joins with a protein complex. Any mRNA with a sequence complementary to an miRNA is tagged with that complex. So look right here. I'm going to try to make it bigger. Right here, this is a regular mRNA, single-stranded mRNA, but it is complementary to the complex, to this microRNA complex. And if it's complementary, then the protein degrades the mRNA. So maybe that mRNA was useful for a bit, but now we need it to stop. So we're going to cut it into pieces and recycle it. Oh, the cells are so good at recycling things. And no matter what, even if the complementariness isn't perfect, it will still block translation. So this might have been an mRNA that was useful at a point, and now it's been tagged, and now it will not be translated into a protein anymore. So that's called pre-translation, and the microRNA interference, all that. Okay, we just have one last thing to talk about, and that's post-translation. I don't think that this is even mentioned in your textbook. This is mentioned in a different version of the textbook. But the idea here is that when we have proteins, just like with the mRNAs, we need to eat them up. We have exonucleases that break them down. We have all kinds of things that cause them to break down. We're going to break down proteins that are no longer needed as well. So there's a particular protein that's called ubiquitin, and it's called ubiquitin because it is super, super, super common. And the word ubiquitous means everywhere. So ubiquitin can tag a protein. So take a look. Here's a protein. Notice it's alpha helices. It gets tagged with ubiquitin, which causes a proteasome, which is another complex that's capable of breaking up the protein and then recycling it. So all of these little protein pieces can be recycled and reused by the cell. So it's a way of getting rid of old proteins that we don't want anymore. Okay, ladies and gentlemen, I didn't realize it was going to get close. There we go. So we are all done. Please let me know if you have any questions. And I'm sure you will have questions because this part was pretty tricky.